So I will present you today the complete genome of lumpy skin disease virus, which was detected in Bujanovac in 2016, and this was detected in Serbia. So this work was done together with my colleague from Serbia. First was Scientific Veterinary Institute in Novi Sad, and then Veterinary Specialized Institute in Kraljevo and Veterinary Specialized Institute in Niš, all three from Serbia. And I'm coming from Slovenia, from Veterinary Faculty, University of Ljubljana, and I am a virologist. I'm working on a special dangerous viral disease like foot-and-mouth disease, African classical swine fever, and also including lumpy skin disease. So next slide, please. So lumpy skin disease virus belongs to Poxviridae family, and in genus Capripox virus, genome is a double-stranded DNA virus, and it is about 150 nucleotides long. So lumpy skin disease virus is closely related to goatpox and sheeppox viruses, and with them share on a nucleotide level about 4% difference. So it's quite close. These viruses are quite close related to each other. So viruses, next slide, please. Viruses which are detected mainly in cattle and are producing huge economic losses in cattle are presented here in the middle, and then another capripox and sheeppox virus you can see on this small phylogenetic tree. On the left, on the right side, you can see the electron microscopic picture of this virus. And because this close genetic relationship in some country, live vaccine against lumpy skin disease virus are used, and this live vaccine can be heterologous strains like sheeppox virus or goatpox virus, but mainly they are used homologous vaccine of lumpy skin disease virus in cattle. Next slide. So the story started in Balkan area. It started between 2015 and 2016, mainly in this part of area where I will present some data. But of course, lumpy skin disease is present in Africa and some east country close to Africa. And disease was spread, introduced into the Turkey in November 2013, and then it subsequently spread to this country. And even though Turkey was started immediately with the vaccination, the disease was spread around to the country in almost all municipality, and later it was detected in Western Turkey in May 2050, close to the border with Greece, which is the first country of the European Union. So from May 2015 to August 2016, more than 1,000 outbreaks of lumpy skin disease were reported from this region, starting first in Western Turkey and then spread to the Balkan countries like Greece, Bulgaria, North Macedonia, Serbia, Kosovo, Albania. Next slide. Here you can see on the right side the map of the Balkan, and you can see the number of cases which were reported in this Balkan area. And you can see also the seasonal influence on the spreading of disease, because this is related to the spreading of the through the vectors. And during the summertime, we can see in 2015 there were much more positive reported cases than in winter times, and the same it was happening here in 2016. Of course, when this exotic disease entered into the European country, mass vaccination started immediately on this area. And of course, because of this, after starting mass vaccination, the number of reported cases was going down. Next slide. You can see here some map from 2080. So in Balkan area, mass vaccination was done with homologous vaccine strain and over 2.5 million animals were vaccinated, and the mean vaccination coverage was about 70%. Here you can see on the map the country which were vaccinated against lumpy skin. Here on the north part, the last country was Croatia. This was preventive mass vaccination, and here is Slovenia, where I'm coming from. So in this area, this vaccination really stopped the spreading of the disease. Next slide. And in 2019, in March, EFSA, European Food Safety Authority, declared that the Balkan area is free now of lumpy skin disease, even though during this time the vaccination was still in place, but now is really stopped because there is no any more report. Yes. The next slide here, I present this first case of lumpy skin disease virus in Serbia in June. This was happened here is in south part of the Serbia in municipality Bujanovac, and of course this is closely related to the North Macedonian cases and Bulgarian cases. Next slide. This is the clinical picture of this first cattle which was affected in this village, Bujanovac. And of course, this cattle was euthanized, sent to the pathology, and we can observe a normal clinical picture of the disease with high temperature, with lethargy, and of course, animals was sent on the pathology. And from this animal, virus was isolated from pathological changes on the skin, on other organs. And this virus isolation was done by Dr. Tamas Petrovic from Institute in Novi Sad. And then after isolation of this virus from pathological material, this DNA was sent to our laboratory for sequencing. Next slide. So sequencing in our laboratory was done by iTorrent technology. So we started use this technology in 2014 in our laboratory, but this was the larger genome which was sequenced during this time in 2016 in our laboratory. Next slide. I will not going deeply into the methodology because probably the majority of you know it. We obtained the complete genome of lumpy skin disease virus, and you can see here from the position 1 to position 150,000, and you can see very nice coverage of all part of the viral genome. Of course, this sequencing was done from extracted DNA from isolate, which we obtained. Next slide. Here are some data regarding the sequencing results. So number of reads were more than 1 million, and mapped reads which were mapped to the lumpy skin disease reference sequence were about 3.5%. The average map length was about 160 nucleotides, and average deep was around 43 point. The total nucleotides which belong to lumpy skin disease virus were more than 6 million. Next slide. So you can see here in the one position, 27,420, that on different part of the genome, some point mutation were observed according to the reference sequence. But nevertheless, very closely related sequence to already obtained strain which is in GenBank was defined. Next slide. Yes, we have some problem with the sequencing with this iTorrent technology, where we have several letters which are completely similar to each other, and with iTorrent technology we cannot define if here is 7 or 8 letters. So we have to sequence such a region in viral genome, and there were about 20 different locations in genome, and we clarified this by Sanger sequencing, and additionally 40 pair, about 20 pair of primers which we designed, according to the previous data which obtained by the ion torrent sequencing. Next slide. Yes, this is the final result. So we obtain complete genome, which consists of 150,661 nucleotides, and this strain is now presented with this number in GenBank. And results of this sequencing was that these strains from Serbia, Bujanovac, have 99.95% nucleotide identity with South African lumpy skin disease virus Nettling Warburg's LEV strain, which is in GenBank with this accession number, and this strain was isolated in South Africa in 1999. Next slide. So you can see here when we compare this Serbian strain to the strains which are now available in GenBank, you can see here that strains from Greece, strains from Turkey, strains from Russia, strains from Kazakhstan are closely related, almost 99.99 to strains which was detected in Serbia. This confirmed that this strain was spread in this Balkan area and also in some Eastern European countries. Next slide. Regarding data analysis, we determined 156 open reading frames. On some position, there were 12 such a position where there were changing in nucleic acid, but there were another 11 position where we didn't obtain any changing of amino acid. I think this have minor influence on this. But we, at the end of the genome, we detect also some deletions, which were detected also in some other strains, which were detected later in this Balkan area. Next slide. So this was published in Genome Announcement in July 2017. Next slide. And this was the first complete genome of lumpy skin disease virus determined in Serbia, but also during this time in Balkan area. Of course, nowadays we have from this region, like Turkey, Greece, and neighboring country, also strains which were sequenced in full genome, and they were completely the same like strain which was detected in Serbia. Thank you for your attention.